This work looks at therapeutic ultrasound's effect on metabolic activity in the rat pancreas slice. This work follows previous work from our lab, which looked at the effect of therapeutic ultrasound on cultured rat beta cells and showed that this ultrasound could induce insulin release from these cells in a reversible manner, meaning that the cells would recover and there was not excessive cell death. Here we observe LIPDH fluorescence, which can indicate the amount of metabolic activity, as LIPDH is in a direct inverse relationship with the amount of ATP in the tissue. So the pancreas needed to be prepared by slicing for these experiments. So figure 1 shows an example or a diagram of how the pancreas is prepared. It needs to be filled with agar to provide stability before being removed from the rat. Figure 2a shows pancreas filled with agar embedded inside of agar to provide stability during the slicing, which can be seen in figure 2b. Figure 2c and d shows the imaging setup with the therapeutic ultrasound transducer. The therapeutic ultrasound transducer is highlighted with a white arrow in figure 2d. Figure 2e, f, and g show 3 wavelengths, bright field at 488 and 633 nanometer, that were used to image the pancreas slice. So the 488 nanometer was used to observe metabolic activity, and the 633 nanometer light can highlight the islet very well, which is shown in figure 2J. So with a black arrow. So that's used to segment the 2 tissue types in our analysis, and also it's very useful. The highlighted islet is very easy to see motion, which can allow us to exclude trials that have excessive motion, as this type of motion is difficult to correct for, since it is random and non-periodic, unlike motion in, say, beating heart cells. So this figure 3 shows the result of an ANOVA statistical test, separating the data based on tissue type and the imaging wavelength. So while we don't see any significant differences with the LIPDH fluorescence, we do see some with the 633 nanometer, which we analyzed since we had the data, although this type of analysis for the pancreas slice is new and we haven't seen before in the literature. But it appears to be different, providing different information than metabolic activity, just given the difference in the differences that we do see. Comparing the experiments with a paired t-test, comparing the 2 tissue types, since each tissue type was present in each experiment, we can see that the one difference that we do see is with metabolic activity in the islets versus in the exocrine tissue with glucose application. That's expected because glucose induces the release of insulin, so we expect there to be a difference in metabolic activity between those 2 tissue types, which is indicating that we are in fact observing a metabolic activity. One reason that we might not have seen the types of changes that we were expecting is because of significant thermal effects that are happening due to our setup. The fact that the tissue is in an imaging well, which doesn't have a large volume of liquid inside. So for the 3-minute application period, a lot of the transducer inefficiency is converted to heat. But we can see that with the pulsed ultrasound, the blue line in figure 5, there isn't a lot of additional thermal energy added with these measurements. Interestingly, when we look back at figure 3, it appears that the pulsed ultrasound, the 800 kilohertz pulsed for all of the experiments, has the closest distribution. So it might be that these effects, the thermal and the mechanical, are competing and perhaps causing the results that we've seen so far. So in the future, we hope to perform more experiments with pulsed ultrasound and hopefully clarify what some of these results mean.